Good afternoon, everyone. Welcome to this afternoon session. The session is forum number 1, Molecular Aspects of DCIS Progression. I'm happy to be with you here today. My name is Shelley Huang. I'm a breast surgeon at Duke University, and it's really my pleasure to introduce an all-star lineup today of physician scientists who deal with DCIS. We will have 3 presentations in total. And at the end of each presentation, there will be a 5-minute live Q&A. So without further ado, I'll go ahead and introduce the first presenter, who is Dr. Robert West, and he'll be speaking to us about tumor stroma and DCIS progression. Dr. West is a professor of pathology at Stanford University, and his research focus has been both on triple-negative breast cancers as well as the progression of neoplasia to invasive carcinoma, where he's made some seminal contributions. He's one of the principal investigators in the Precancer Atlas, Human Tumor Atlas Network. Thank you very much, Dr. West.
Great. Well, thank you so much for inviting me to talk here. I really appreciate the opportunity. I'm going to hopefully give the audience a little bit of an idea of the tumor stroma in DCIS progression and talk a little bit about the work that we've recently done within the context of the Human Tumor Atlas Network and the Precancer Atlas. So I'm sure this slide is quite familiar to everyone, but ductal carcinoma in situ increase has increased with screening. The DCIS, of course, can be treated similar to invasive breast cancer with a combination of different modalities. However, we can recognize that actually it's only a subset of those DCIS that really would benefit from this aggressive treatment. So the question is, can we study DCIS further to identify cases that might benefit from less aggressive therapy? And of course, the topic of my discussion today is the tumor microenvironment. And I wanted to quickly point out some of the differences between invasive breast cancer, IBC, and ductal carcinoma in situ in terms of the tumor microenvironment. Of course, invasive breast cancer is intimately associated with the stroma. The cells are in direct, the lesional cells, the neoplastic cells, are in direct contact with the stroma. They've escaped the basement membrane confines. And of course, they interact with fibroblast populations and immune cells, among others. In contrast, while DCIS also interacts occasionally with fibroblasts and immune cells, there are other cells and features to consider. Of course, in the breast, there are the myepithelial cells, and those are playing a key role in organizing the epithelium. And in addition, there's more of an epithelial compartment organization, with including things like hypoxic gradient from the periphery of the DCIS duct into the center, where it can be quite hypoxic. I want to also note that there have been a number of really seminal studies that have examined the stroma in invasive breast cancer. And this is a study done by Nellie Pollack's group back in 2004 that looked at identifying individual components and their expression factors in invasive breast cancer. Dr. Park's group also did some seminal studies looking at stroma in predicting outcome in invasive breast cancer. And then more recently, there have been proteomic work, and this is a terrific paper by Raza Ali's group that looks at a number of genomic and cellular features in the tumor microenvironment, analyzing it with proteomic type approaches. Okay, but despite the fact that there are differences in DCIS and IBC, of course there are similarities, and so we want to extend the observations made by others and understand what's going on in DCIS to better risk stratify those cases. Of course, there are a number of new technologies. I've already mentioned the proteomics, but there are new spatial transcriptomic technologies that use a variety of sophisticated techniques to either dissect out important pieces of tissue like the stroma, or do in situ analyses, multiplex in situ analyses, to look at the stroma within the actual tissue. And of course, there are a variety of multiplex antibody-based imaging methods, including what I'll talk about today, which is the multi-ion beam imaging on the far right, designed by Mike Angelou, who's one of our collaborators. So the study that we did was to look at two different cohorts, the TBCRC cohort and the rabbit cohort. Both are retrospective cohorts with long-term outcome where we understand which patients don't develop any ipsilateral breast recurrence and which patients do, either with a DCIS recurrence or progression to invasive breast cancer. And we looked at a number of different modalities. And we did this by taking multiple sections, serial sections, of the archival paraffin-bedded tissue. And then each slide then goes off to make either an RNA study, DNA study, or the proteomic MIBI study down below. So first I'm going to talk about the MIBI work. And this is, of course, using multi-ion beam imaging, or MIBI, which involves using a set of antibodies that have been individually tagged with different metal ions. And that set of antibodies can be simultaneously staining a single slide of tissue. So basically we'll get a panel of 30 or 40 different antibody signals on a single slide. And this is an example of that, where we can see that there's a variety of stains. Here we're just showing some stains for immune markers and keratin markers and stromal markers. And effectively, the panel that we used is a 37 marker panel that can identify over 1,000 different tissue features, both cell composition, functional subset, and spatial features. And this effectively digitizes the tumor microenvironment. So in the past, we've been reliant on pathologists like myself to do annotations of the tumor microenvironment, identifying, at least in this case in the middle panel, the epithelial compartment in white and the stromal compartment in black. But we can now also do that using the MIBI data with computer annotation. In fact, we can even refine that to identify the stromal, the epithelial, the myoepithelial, and even sort of center luminal compartments where there might be more hypoxia. And of course, we can identify a number of different cells. On the right-hand panel is sort of the variety of different cells, both immune and non-immune cells that we can identify. And using this population, this identified population, we can start to find discrete tumor microenvironments. The first thing that we did was to identify cellular and structural features associated with a change in progression. That's to say, going from normal breast to DCIS to invasive carcinoma. And this heat map in the middle demonstrates a number of important pathways that are either upregulated or downregulated as we make these transitions. But more interesting is that we can start to look at predictors of invasive breast cancer relapse. And this is using one of our cohorts, the rabbit cohort. And in this case, we could look at both spatial and non-spatial features. That's say spatial features, the relationship between cells and different compartments or other different types of cells. And non-spatial features, perhaps the expression of what's in those cells. And in this study, we identified a number of different features that predicted invasive breast cancer progression. Here what we're showing is the top 20 features. And it's heavily predominated by stromal and myoepithelial features, as you can see here. In fact, the top feature on the left-hand side is the expression of E-cadherin in the myoepithelial cell population. And finally, the spatial organization is a crucial determination of risk. Sorry, just advance here. Right. The phenotype and structure of myepithelium here is very important, and we identified two different types of myepithelial phenotype. One is this continuous myepithelium that we can see here on the left, and then another group had a thin or discontinuous myepithelium. And I think when we first observed this, we thought that it was likely that the thin discontinuous myopithelium would be associated with an increase in invasive breast cancer risk, because that so-called barrier is breaking down. But in fact, it was the opposite. It was the discontinuous myopithelial layer that was associated with less risk. And the thinking here is that actually the myopithelial thick layer actually protects the DCIS cells from the immune system and the potential control the immune system could have on the neoplastic cells and their ability to progress. Whereas if the myopithelial layer is breaking down and allowing immune cells to enter into the DCIS compartment, that allows the immune system to start to control the neoplastic growth and perhaps put some brakes on the progression process. Okay, so that was the MIBI analysis that we did. We also looked at RNA expression both in the epithelial compartment and the stromal compartment using laser capture microdissection. And in this case, we used a case versus control analysis, starting out with the TBCRC cohort. And in that cohort, we identified 812 genes that correlated with risk. And here we're showing the Kaplan-Meier curve showing that in the original training set, of course, we can see that there's a big difference in high risk and low risk DCIS outcomes with respect to both invasive breast cancer and DCIS. In the validation set, the rabbit set, we can demonstrate we also see this risk as well. And if we look at the hazard ratio comparing this classifier against traditional clinical pathologic variables such as the treatment, age, DCIS grade, we find that actually the classifier here is the only significant event in this multivariable analysis, both for the training and the validation set. We also did a number of other analyses to examine the biology of DCIS, and using both these data sets, we were able to identify 3 strong subgroups of DCIS, an ER low, an ER high, and a quiescent subgroup. And these subgroups had their share of the drivers of that 812 gene risk classifier sequences, genes. So on the far right there, we show the number of gene pathways that are significant in the 812 gene classifier set and how they're differentially expressed in these 3 different RNA-defined subgroups. Finally, we looked at the tumor microenvironment itself and found 4 different subgroups of tumor microenvironment based on purely RNA expression. And these subgroups, again, on the far right-hand side, demonstrated a number of different pathways associated with risk. In fact, in our risk classifier, 812 gene classifier, about half of the genes involved are stromal-based genes rather than epithelial-based genes. So the stroma and its interaction with the tumor is playing a huge role in controlling the risk of recurrence for the DCIS. We were also able to go back and relate this to the MIBI data, the proteomic data that we had. And of course, remember, we cut these, the tissue used for these different modalities was cut from consecutive slices. So in effect, we're looking at the exact same ducts that were studied by MIBI as were studied by RNA. And in this slide, we're demonstrating that we can find the stromal phenotypes associated with ECM are present in this breast cancer progression. And so we can identify collagen phenotypes, the length of the collagen fibers, the organization of the collagen fibers, and we can relate those to the expression patterns of the myofibroblasts. And finally, in panel E, we can demonstrate that these different cell types have a variety of different extracellular matrix proteins that are expressed. And then finally, I wanted to just note that the studies that we're looking at are, because of throughput issues, really focused on individual regions of interest within a patient's DCIS. But of course, patients often present with multiple centimeters of DCIS, we're not accounting for the heterogeneity within those lesions. This is a study from Yin Wang's group, who looks at H&E and makes observations on H&E for cell types and the presence of different cell populations. And this kind of work on a much larger scale, which can then start to look at heterogeneity, I think is really going to be a key in approaching these issues and creating a more holistic output of the entire patient's DCIS. So, in conclusion, pre-invasive neoplasia microenvironments are different from invasive microenvironments in a number of key aspects. The new approaches, in part what I've presented today, can start to address the spatial issues, the expression issues, and give insight into biology. And finally, we've identified gene pathways and spatial features that can predict outcome and hopefully can ultimately be used in the clinics to help guide patients that are initially diagnosed with DCIS. And then the other point I wanted to make is that I think the clinical endpoint is important in the study design. In these studies that we've presented, we've looked at either the recurrence of DCIS or of invasive breast cancer, but we haven't asked how aggressive that invasive breast cancer is and whether upon having a new diagnosis of invasive breast cancer, whether that patient would need systemic therapy. So those are important questions to address in future studies. The two papers that I talked about today are published, one in Cell with Tyler Rysam as the senior author, and another in Cancer Cell with Sari Strand as the senior author. So I encourage you guys to look at that. And then finally, I wanted to leave you with this thought, and that is we're looking at patients with a diagnosis, a clinical diagnosis of DCIS, but that's really the tip of the iceberg in terms of the spectrum of DCIS disease. If we think of DCIS as a time that the clone, the neoplastic clone, is dwelling at the DCIS stage before transitioning to invasive carcinoma, then the DCIS that we've studied today are really high-risk and low-risk DCIS well on the right-hand side of the spectrum of DCIS dwell time. In fact, on the other side is DCIS that presents with invasion. Those DCIS have a very short dwell time, and we missed those patients on screening. So those are the patients, if we could study those patients, that would have the biggest effect. And then finally, I want to acknowledge this is obviously a multi-institutional study of which I'm a small part of. And this is really a collaboration between researchers at Duke, Washington University in St. Louis, Arizona State University, and of course, folks at Stanford, and Dana-Farber, and the London Institute of Cancer Research. Thank you very much.
Thank you, Dr. West, for that presentation. I'd like to invite anyone with questions up to the microphones. And for those of you who are attending virtually, you're welcome to put your questions into the chat. But Rob, maybe I could just start off. You know, I think one of the questions that are probably on everyone's mind is, you know, this is yet another DCIS classifier. And so can you tell us about your thoughts of how this will compare to the other classifiers that have been out there in clinical use for the last few years?
Yeah, that's a great question. I think perhaps the main difference is that some of the commercially available classifiers are based on patient cohorts that were relatively old, and I think the clinical treatment of DCIS has changed to some extent in that time. So I think the patient cohorts we're looking at are more modern. The other, though, important feature is that really we're studying the DCIS biology itself. Some of those other classifiers were adopted based on observations of invasive breast cancer. And so I think it's really important that DCIS is living in its own tumor microenvironment, the intraepithelial compartment. That's a very different compartment than the environment that the invasive breast cancer lives in.
Yeah, thank you. And we have time for two questions. Maybe this person in the center, I think you were first.
Okay, thank you. I'm Jose Ales from Spain. I didn't see, perhaps you mentioned it, are there differences between the things that you see among HER2 positive, HER2 positive, I mean, ER positive and triple negative DCIS?
Yeah, so does subtype influence all of this? So there are features in DCIS that correlate with invasive breast cancer. And you mentioned the subtypes. There are also copy number changes that are found in DCIS that are present in invasive breast cancer. Some of those copy numbers are associated with invasive breast cancer. However, when we looked at the subtypes and we looked at copy number alterations, those did not correlate well with the outcome differences that we're observing in DCIS. So those may be influencing the biology of the invasive breast cancer, but perhaps not as influential in the biology of DCIS progression.
Great, thank you. And one last question over there, please.
Thank you. Ranju Gupta from Pennsylvania. So a couple of years ago, we were talking about overdiagnosis and overtreatment of patients with early stage breast cancers, perhaps the DCIS and the LCIS. Do you think with these modern classifiers, we would be able to define those patients who perhaps can be just observed because they have that low risk DCIS, which will not progress into invasive cancer, and we can leave them alone? Like the analogy would be like prostate cancer, right? I mean, we don't have to treat everybody.
That's a great question. And yes, absolutely. I think that would be one of the main goals of this work, is to identify patients that would benefit from not as intensive therapy. And maybe that's some sort of watchful waiting approach.
Thank you.
Yeah, thank you.
All right, thank you so much,
All right,
Dr.
thank you.
West.